And so I'm just going to introduce our first speaker, Professor Goense. So I think we're really fortunate to have Professor Goense giving this lecture. I think that there's not many people who have experience both in electrophysiology, sort of hardcore neurophysiology, and laminar fMRI. So I think she has a really unique perspective to offer us, and we're really happy to have her here today. Thank you, Jozien.
Thank you very much, John, for the nice introduction. Thank you for coming here and for inviting me. And I feel honored to be the first speaker in the first laminar fMRI course. So I'll be giving both a general introduction and talking about the neuroscience behind it. So we're gonna it's focused quite a bit on the biology, and we're gonna go into the neuroscience quite quickly. So feel free to ask any questions, but be aware we're going to it what is relevant for the for laminar fMRI specifically. So what's the questions is I have divided it up is in why do we do it? Why do we want to do laminar fMRI? How do we do it? And what are we measuring? And what do we want to know? What are the questions we wanted to answer? So why it's the promise of laminar fMRI? There's of course great promise because the cortical layers have different cell types, different function. They constitute the cortical circuit. They have different connectivity. So that gives you a handle on mechanistic questions and on cortical circuitry. So and that is like what regular fMRI has to a much lesser extent. It really can answer mechanistic questions. The how, here we are going to go over very quickly because a lot of people will be talking about this. And then the what, because it is a complicated signal, as John has already mentioned, with many, many aspects. So, and that's why this talk packs a lot of information in an hour. And we're going to correlate it with some other neurophysiological metrics. And then finally, what can we learn from it? Because in the laminar fMRI literature, you also run into this question, what is the ground truth? And whether it's neurophysiology actually the ground truth, or what do we compare it to? So we'll talk a bit about that. So that's what we see here. Oh, that's quite purple. So we have the V1, the laminar cortex, in striate cortex in V1, and that you see this is a Nissl stain for the cell bodies. So you see a lot of differences in the layers in V1. You can also see that in the MRI signal and in the high resolution fMRI signal. We hear at about 200 microns. It's an animal system, so that these resolutions are achievable, even though still challenging. And as I said, we can have access to that cortical circuit, and that's what you see here. So this is V1. V1 is the model system of the cortical circuit in that sense. So a few of these models have been proposed by neuroanatomists and neurophysiologists, and some of the most well-known one is the Douglas and Martin one, and that's the one here. I think they come up with it. So that says that basically the input comes in from the thalamus to the granular layer, which is layer 4. From there, it's further processed in the supragranular layers, 1, 2, 3. And then you have that feeds back on the infragranular layers, 5, 6, and they send output to the subcortical structure. It says thalamus in here, but it's actually several subcortical structures. Another very well-known model is the excitation inhibition model. That is like balanced excitation and inhibition. We won't talk a whole lot about that. If you do spectroscopy, you have probably a bit better access to that. And then there's the connectivity model. That's here. That's the Velleman and Van Essen model that's very well known for most people. And that says that the connectivity is quite segregated. It's a canonical model, so it's largely true for most areas, but every brain area is different, and it's not universal, exactly the same all the time. But the key connections is the input into layer 4, and then that's the forward input, and then the feedback input to the upper and to the deeper layers. and also they originate in different places. So with laminar fMRI, we've had some successes. We can see now, we have seen the cortical layers, and you can see that here, you saw it in the monkey V1, that we really can distinguish these layers, the two layers here. And that was already quite an achievement, because initially when I started out, they were debating what is the point spread function, like 1 or 2 millimeter, which means you could see nothing. So we have really established in this time that we can see that it's feasible, and that you with different stimuli, you can also see different laminar patterns. Now, how to interpret them, that's a lot of it I'm going to talk about now. So I'll briefly introduce the model. One of the main models, I work on rats and monkeys, but a lot of the early laminar work was in monkeys. And that's the macaque. The macaque has the advantage, especially the V1, the visual system of the macaque, old world monkeys, is very similar to that on human, and especially early in the visual system, the V1 and early visual cortex. And that's what's seen here. And that's like here you have the cortical expansion between a human and a monkey or a macaque in this sense. That really the massive expansion is in temporal, frontal, and temporoparietal, which is tools, language, and all, while the primary visual, primary sensory has seen much less expansion. So the macaque's a really good model for human vision. The area is mostly, I'll talk a bit about other areas, but mostly I'll talk about V1, primary visual. We've already seen the cell body stains, the Nissl stains, and the rare, where you can see the cortical layers in V1. You can also see the vasculature. You see here the layers, and I think you can see here the blood vessels. We'll come back to that. But V1 in monkeys has been very well studied with all kinds of methods, and it's been the electrophysiology model of choice for decades. So this shows the cytochrome oxidase stain, and that's layer 4. And you see that layer 4 has much higher energy use. Cytochrome oxidase is an enzyme. It's the final step in the oxygen metabolism step. So any area with high cytochrome oxidase is thought to have high energy consumption. And we'll see the columnar structure, so I'll come back to that. So I'll talk mostly about biology. The fMRI comes a bit on a side note in this talk, but we go from the issues. We have cells that differ in the layers, we have connection difference, we have the cortical circuits. We also have different neural signals. So we have energy use, we have the blood flow, and all of these pack into that fMRI signal. So what do we compare it to, and how do we interpret our results, basically? So I'll briefly go into the technical stuff. So the technical issue, the major issue is, as John has said, we basically don't have the resolution to see individual layers. Maybe in some cases, if you have 200 and your layer is 600, like V1, but for the most part, we don't have the resolution, so we tend to see a bit of mixture signal, and that's what's shown here. So 500 microns or half a millimeter was my standard resolution, or my starting resolution for monkey work. But if you see layer in V1, you have 4 voxels that span the whole cortex, and that's what you see here. Layer 4 is about 1 voxel, and then you have supra, infra, and then layers. In humans, we have 750 is now fortunately pretty standard, so which is three voxels, which covers just covers layer 4, and so we have 3 voxels to span the cortex. But then we have also this vascular layer. I'll come back to that, but you see that there's a large cortical vessels in monkeys, about 1 millimeter already, or on top of the cortex. So because V1 and S1 also are only 2 millimeters thick, a lot of the early work, of course, is focused on M1, which is much thicker, that you can see here. There's in the somatosensory cortex S1, and here's M1. So you see the difference in thickness, and you also see these massive cells in layer 5, the bed cells. And it is shown here schematically again. This is primary visual cortex with the thick layer 4, and then primary motor cortex. So actually it's not to scale for thickness, but if you see V1 and M1 are actually pretty unrepresentative parts of cortex. The V1 with the striation, high energy use, high blood flow is quite an atypical cortical area, and M1 also. M1 has very thick layer 5, 6, and barely any layer 4. It's a long time there was a discussion whether it was a layer 4 at all. And this is much more typical. So why M1? And unfortunately, M1 is not so much a laminar model for animal work, and the reason is mostly the animal has to engage in a task, which is difficult, and in MRI you have, of course, motion issues. There's a lot of interesting work done with mapping. So there you have the monkey doing a task while being recorded, and then they have these arrays that there's some classic work by Martinos and all on plasticity and finger mapping that's done at high resolution at 7 Tesla now in humans, but it's also done in monkeys with arrays, and then the mapping and remapping after injuries and all. But laminar work has not been so much done, also partly because of the technology in neurophysiology. And of course, it would be nice, you would have to have a scanner like this, which is some of the work is on this scanner. It's a 7 Tesla upright, but I don't think that one is in commission anymore. So it makes doing M1 work quite difficult in animals. Another thing we have to consider is there's now an explosion of the work in mice, basically. But there's many reasons for. One is the genetics models, and the other one is the optical techniques that are so widely available now. So this shows the picture of the monkey visual cortex, and here you have the mouse. So you already see there's quite a large difference with the layer 4, much more thinner here. And that's shown here schematically. The difference between species is all the primates have pretty much thicker cortex than the other animals, rodents, carnivores. And that's what you see here. Especially the mouse has 1 millimeter cortex. Bad for us, but it's very good for the optical folk. And if you scale it, you also see that the cortical layers, the laminar structure is different, and the largest difference is that in the primate, the layer 2, 3 has expanded enormously compared to other animals. So that's really what the primate line has that's very different, is the layer 2, 3, and that's of course the cortical processing. You see that here in the rat. You can see that layer 4 is here much more to the surface compared to what you saw in the monkey. So it's not only the spatial resolution, but you type into the scanner your nominal spatial resolution, if that were only it. But there's many other aspects that often degrade your resolution, and that you have to take into account. So there's sequence type specificity. This is just an example, gradient and spin echo. So they have different patterns. You have the parameters. That's also in this paper. Not only is your sequence important, also the parameters that you set. Then you have distortion. You have an effect of your analysis parameters. You have which metrics, and that you can see here. If you use a percent change, you get suddenly a very different pattern than if you do a correlation coefficient or a t-test, especially for this gradient echo. And then you have a lot of analysis, establishes potential for blurring. You have motion, but all the many other people in this course will be talking about that. So I'm just mentioning it as an introduction, and that's it. I will deal with the biology. So what are we measuring? So we know the BOLT is related to blood flow and to metabolism. So here we have the anatomy, as you already saw briefly. This is the corrosion cast of the monkey V1. And it shows the capillary bed. This is the thickness of the cortex. If you see, the white matter is much less vascularized than the gray matter. You also see these vessels. These are the arteries in red, veins in blues. These are the penetrating arteries and vessels and veins. And then you see the large vessels that lie on top of the cortex. So, and that you can also see in the MRI image in the anatomy. So you see basically here's the large vessels. This is the distortion effect from the large vessel. And then you see the penetrating veins here. And you can also see it in a human. And you see, here you can see the large vessel that are overlaying. And then you see here the veins. It's not that in the human, the higher resolution you go, the more of these veins you see. It's not that there's fewer in the humans, you just need a bit higher, at higher resolution, you're going to see more. And as you all know, the BOLT signal is a measure, it's a combination of flow and metabolism, because you measure the deoxyhemoglobin, so the metabolism, oxygen consumption comes into it, and then you have the overshoot by the blood flow. But at the cortical laminar dimension, if you saw the vascular anatomy, what you also have is that the blood goes like that. So whatever is in the deeper layer has first been in the superficial layers. And that's something also to keep in mind. But the other question is if you do laminar fMRI, which laminar fMRI signal? So here's in a monkey, we did in V1 again, multiple different fMRI techniques. So we have the gradient echo BOLT, which shows the profile that's calculated like that and like that. It shows the profile of the activity mostly on the surface. That's what many of you are probably familiar with from gradient echo BOLT. The spin echo BOLT shows this profile with more activity in the middle. CBV has this double peak in V1, and then you have CBF and you have CMR2. So CMR2 and CBF are more focused on in layer 4. And in this case, I overlaid it on the cytochrome oxidase picture, and it's quite nicely in agreement. So if you see the vascular properties, they also again differ in the cortical layers. So this was again the corrosion cast, but if you do the quantification of that, you see this is the total vascular volume. And you see again that layer 4 is more vascularized, this has a higher blood volume. And I hope this green comes through, that agrees with the same pattern is seen in the cytochrome oxidase. And that's what you also see with MRI. Here we injected the contrast agents. You see really the different layers already in the cortex. So white is higher blood volume. So you see that layer 4 has a higher blood volume, and that's what's seen in the R2 star profile. And that agrees quite well with this profile. So you see there's difference even in blood volume within the cortex. A further question we can ask, is the neurovascular coupling the same in the cortical layer? So that was the focus of an earlier paper, where we did compare positive and negative BOLT, and you see again these 3 different patterns for BOLT, blood volume, blood flow. But in the negative BOLT, instead of seeing the inverse of the positive BOLT, you suddenly started seeing a different pattern, and you started seeing that the blood volume increased while the CBF decreased. So It's quite a complicated response, and that also suggests that the neurovascular coupling might not even be the same in different layers. And that's here just given schematically for positive versus negative pulse. So now what can we do? I'm going to make a detour of the columns, and well, many people who do laminar fMRI do columnar fMRI as well, but also because the columns help us to understand the circuitry. So this is basically the cortical columns that with a schematic, with the neurons, with the dendrites, axons reaching through the cortex and then going into the white matter. Cortical columns tend to have the same response properties as opposed, like say, if you have an orientation columns in V1, and this columns of neurons responds to motion in this direction, or basically, or even a bar in that direction, then the neighboring neurons will respond to a bar that's like that. And that's, you can map that as orientation columns, as you see here. For each color has a different orientation of the bar. This goes back to the Hubel and Wiesel work who did that in V1. But each column has a different orientation. But within a column, the response is the same. And the same you can do for an ocular dominance column here in a monkey. It's just the neurons in each column respond to one eye, and then they switch. So the neurons within the column tend to have a lot of the response to the same thing. And these have also been shown with fMRI, here in this case in the cat, with the CBV method. You can see if you subtract two orientations, you can get these columns. And the same in the human. This is an ocular dominance column, and here we can see the orientation column. So you do the different orientations. But columns are also in other areas, like in this case we have the temporal lobe. This is a monkey, and we see that in different columns, or sometimes in high areas, they tend to be called patches also. They respond to the same stimulus. So, and then in this case, apples or the monkey faces. And you can see this with optical imaging. This is optical intrinsic imaging. But you can also see that with fMRI, you have this patchy structure in the temporal lobes. So another model that's very, very well known in neuroscience is the rodent whisker barrel cortex. And that's, if you see here, each whisker is represented against in that columnar fashion, like here. Each whisker has its own spot. So you can have this whisker barrel on the cortex. And that's what you see here. You have the C2 to say A1 or A to A5. And then all these whiskers are labeled. Every rat is the same way. And you can see that here. And you see how small they are, because this is the overlying veins, arteries. So that's already says, okay, how that can tell you about the regulation of the blood flow. And that's what this picture shows. So if you were to measure only large veins, you would never be able to see these whisker barrel columns. But here they have done optical imaging. You just move one whisker at a time, and you can get this columnar pattern. And that corresponds again with the cytochrome oxidase response in the. So if you see that for a single whisker, you see here this pattern, and you see the response to one whisker, and it shows it very nicely aligns with the column. But if you see, these are the labels of the veins and the arteries that dive into the cortex, and you see that the spacing is larger than the column. So this is only possible if you have a capillary response. So the columns in that sense tell you that the regulation is very fine, and it's at the capillary level. So if you can measure capillary responses with fMRI, that means you can see this kind of very small features. Even if you measure the penetrating veins, you won't see these small features. So it's really, you have to measure the capillary responses. Fortunately, fMRI can do that, but with the right adjustments. but it is a challenge. So if we now go to a bit deeper into what the cortex actually looks like, we see these authors have done, they counted every neuron in a red barrel column, in one vibrasaur, in one whisker. And you see here's the cell bodies, and they color coded them. So they color coded them and assigned them a layer. So if we say a neuron in layer 5, it means the cell body is in layer 5. It doesn't mean necessarily the rest of the neuron is in layer 5. It means the cell body is in layer 5. Because we know neurons have dendrites, and that's here also color coded, and we see they extend through the cortex, and you see that the color coding is, but it's still confined to a layer quite well, but you start seeing that it crosses layers. The axons is quite a mess, basically. It just goes everywhere. And if you look from the top, we see cell bodies, dendrites that reasonably confined, and axons. Even though it looks pretty messy here, it's still not too badly confined, but it does spread out. And that's again what we see here, that the properties of the cortical columns are not totally uniform. They are, like, say, one column, the neurons will respond to, say, one direction or one broad orientation, but they're not totally the same. And that's what you see here. So the response properties and the receptive fields, they vary slightly in different layers, and this is an fMRI study. Also, and we come back to that, the LFP, the neural response, the spread of the LFP differs a bit in the different layers. So the complication with layers and with laminar fMRI is basically, it's not so neatly, if it's just a cell body, you can say, okay, we see that response, but we see neurons span different layers. And many neurons, especially the layer, this is an example of layer 5 neurons, they go, the cell bodies in layer 5, but they go all the way up to layer 1. So this again, the question is, what are we measuring? So the cortical circuit, and this has been known for a long time. And we saw here's the layer 5 neuron, and this is and these large neurons also found in layer 3. These are the pyramidal neurons, basically because of their shape. And they are often what we record with electrophysiology. There's also these more rounder neurons, stellate neuron, granular or Martinos cells. So these are smaller, but we tend to not measure them as much with electrophysiology or with EEG. So this was always the assumption, if you do an fMRI experiment, that it is equivalent, you say we measure neural activity. So, but that was the implicit assumption, was always like, okay, the fMRI response is correlated with neural spiking. Once, if you show the monkey a face and you record the neurons, you get spiking responses in these, if you record in these areas that are activated by faces. So if you see, what do we compare to? What we record with electrophysiology is basically you put a electrode, a sharp electrode, which you stick next to a neuron, in this case, the pyramidal neurons. Because the pyramidal neurons are large, they're the most likely ones you record. So this is the pattern what you get, and usually they filter it out so you get spikes, actual potential, the neural firing. More recently, more people look at the LFP because this is the actual signal you get. If you filter it, you get spikes, but if you do the low pass filter, you get these slower fluctuations, and these are what you measure with the EEG. And also now the people who do electrode recording with microelectrodes are starting to pay more attention to the LFP. So with the EEG, this is what you measure. I said you measure the pyramidal neurons, but they call what is called an open field arrangement, which means dendrites on the top, and here cell bodies that creates a dipole, and those are the ones you measure. So that's why also around the stellate cell you'll not measure so much, because there's no dipole, it's symmetrical. So here this is what you measure with the EEG. Also with LFP, you tend to measure that. And moreover, it is synchronized. So a single nurse, you just measure with EEG, you measure if they are synchronized. Which goes back to the early question of that Nico was asked in 2001, is what do we measure? Do we measure spikes? Do we measure LFP? And he did, of course, put the recording electrode in the cortex, and he said, okay, it tends to be, everything is correlated, as it usually is with neuroscience, but it tends to be the LFP. And the LFP is thought to represent more. Nobody actually really knows what it represents, but it tends to be more representing of these dendritic events and less of the action potential, but actually there's also a bit of action potential in it. So that's what was discussed quite a bit, like 10 years ago. This was very much in the attention. It's going to be a bit quiet here, but the increased interest in fMRI, but it's still not known how to interpret it. And here another one, Nature Neuroscience, the exact link between fMRI and neurophysiology. And now we do laminar fMRI. So we basically added an extra dimension to that problem, because we don't look now what's the situation in the different layers. And that's with the recording. The recording technology has also advanced a lot. Nowadays, it's much more common to do these laminar recordings. In this case, this is a 16-channel recording, and you see the dots here are the spikes, and the line is the LFP. So this again comes back to that question, is the ground truth? What are we measuring and what are we comparing? So if we say neural activity, because of the things inherent in the recording, you tend to often say pyramidal cells, because those are what you record with a microelectrode, and those are what you record with the EEG. And this is here a schematic of the different ones. So these are the layer 5, these large ones that go from layer 1 to cell body in layer 5. And then the others, they extend also, but they don't extend as much, but they do extend throughout the cortex. And most of us have been learning, this is as a neuron, that's what a neuron looks like. Kind of, you have the myelinated axon, you have the cell body, you have kind of relatively small synaptic, the synapse, and you have here the dendrites. And if you also see, you have all the cell organelles, they were in the cell body, and then the mitochondria specifically draw your attention to, because those are the energy factories. So, but this is a motor neuron in the periphery. It's not a cortical neuron, but because this is what a cortical neuron look like, or especially those pyramidal neurons. Not only have you this nicely divided dendrite tree, you have here this apical tuft of the dendrite, and then you have the basal dendrite. So you have dendrites, one neuron has these dendrites in layer 2, 3, and in layer 5. And then you see here, the axon is very thin and also goes everywhere. But one of the things we see also is, this is also a pretty recent study, that you see where's the energy being used, because as we fMRI people, we measure flow energy. So where's the energy being used in the pyramidal? So we learned, we always think mitochondria is in the cell body, so, and indeed that's where they are, but you see the mitochondria is also all through the dendritic tree, and they are in the axons. So that's what you see here. And here's a enlargement of this and that, where you see that it's stained for mitochondria, and throughout the dendrite you see mitochondria. In the axon there's also a lot, but they have a different shape. That was the topic of this paper, and I don't go into why that was, but you see both mitochondria in both axons and in dendrites. And if we see what cortex looks like, we see this is an A&E stain, this is a standard pathology stain. And you see here's the neurons. Some of these, this is pyramidal neurons, they're quite big. They can be like 40 or 50 microns for the cell body. Here's a capillary. here is astrocyte and oligodendrocyte, the small ones. But what you see most of it is so-called neuropil, and it's synapses, dendrites, axons. So that is what most of the brain actually consists of. It's not so much the cell bodies are pretty, not even that dense. And this is a stain, a Nissl stain, where you also see this, also the oligodendrocytes, the astrocytes are much smaller. And here's a reconstruction where they reconstructed every neuron in a very small patch. It just is nice to show you, and this is 10 microns to how dense it all is. So this is all the cell bodies in the neurons in that size, and this is all the axons and dendrites. So you see it's packed with axons and dendrites. So I go back to the connectivity. So knowing that these axons and dendrites are so important, what does this foraminal-evanescent picture look like? So if you see input, indeed, it comes in layer 4. It's also a bit like layer 3 as well. That's the thalamic input. And there's another thalamic input, but it's much less. It's also much more recent, also layer 4, but also layer 1 and 5 is a secondary thalamic input. And then you see the descending input, that this is the feedback, mostly deep layers and a bit in the superficial layers, in layer 1 specifically. And if you look at output, then they talk about location of the cell bodies. We see that the the output to the higher cortical area, this would be the feed forward, the green one, comes from superficial layers, 3B specifically. And then this is the feedback, and it originates in the superficial and in the deep layers. And this is the output to the subcortical structures. So as you see, not so much seemed to happen into layer 2, 3, but that's all intrinsic connections within the cortex. And that difference is because of, if you do tracing studies, they tend to either label cell bodies or the synapses, and that's just because of the way the traces are designed. So they either go retrograde and measure the cell body, or they go anterior grade and manage the synapses. Very few traces cross the synapse. So that's what you see here. This is the thalamic input in the barrel cortex. So you see, this is the synaptic signal in layer 4, and this is that one. And you see it here. This is the synaptic signal, and here are some very small pink dots, which you can see. These are the cell bodies. So then this field is now growing enormously, but there's a lot of new traces, and then they can actually label specific cells. So it's really a field to watch, is like if you're interested in the connectivity. But you see also that layer 1 has this, a lot of the synapses occur in layer 1, because layer 1 we kind of forget about it because there's no cell bodies there. But we see this, it has a lot of synapses. And that is shown here, if I can start the video. That then, can you start the video?
Oops. Yep. Now it keeps disappearing.
Well, we didn't. Okay, so we see this is in monkey cortex. We go to optical imaging, like to see what it looks like, and so we go through the cortex, and this was the first was the PIA, but now we are in layer 1, 36. Where are we? I want to stop the video.
Yes. It's funny, this trackpad is tough to use, huh? Here we go. I just use the left side.
Left side? Okay. So if we see here, We are at the depth of 50 microns, so you see these spots, they are the dendrite axons and synapses. Here also, we see, and now we start, I'm going to do this by hand. So you see here, this is cell body coming, but there's very, very few cell bodies in layer 1. And so now we go a bit deeper, and here the cell body starts to become really dense. So we're in layer 2, 3. And so you see that layer 1 is very, there's not much cell bodies, but it's very dense. Also the vasculature is also, the capillary bed is also in layer 1. So there are most likely energy use, but what do we see? Then this is a reconstruction of all these dendrites. So we see also that the dendrites arranged by cell body. So we see that, no laser pointer. We see that in the upper layers that the dendritic fields go more up. In layer 5, you see the dendritic field goes all the way from layer 1 to layer 5, and then in the deeper layers, the dendritic field is a bit more confined to the deeper layers. And the black here shows the axonal field. So the red is where is the most of the dendrites concentrated, and the black says where is most of the axons concentrated. So that you say this was a rat where they counted every cell, but same as in humans. That is the temporal lobe in humans. And you see also, the apical tuft that goes all the way to layer 1 sometimes. And then you see the basal tufts of the dendrites that stay more in the deeper layers and more localized, and then the axons go everywhere. So what do we measure? Now, if we go to laminar electrophysiology, I've already mentioned the LFP. You have multi-channel electrodes, so you get these 15 to or 16 to 30 channels of LFP signal. This is the power, what you see. So most of the power is in these low frequencies. The most of the power is in 10, 20 hertz, and then in the higher frequency, that's the gamma, is much lesser power. And that's what you see. Sometimes you don't see these kind of images a whole lot. You tend to see in neuroscience publications, you tend to see more this kind of images where they normalize the baseline activity out. So you see the change is in the upper layers, and you see gamma activity, 40 hertz, that's gamma activity. So these are the bands that you hear people talk about. They come from anesthesia. Delta, theta, alpha, beta, and gamma. What we often hear about is alpha and beta, that's 8 to 30, and then gamma, that's because that's thought to be associated with neural activity, with spikes and feed forward. And then the multi-unit activity is spikes, tends to be above 300 hertz. But you see, oftentimes you have relative differences. If you see also the LFP, what do you see? So we have the action potentials to the right, and then the red is the action potential. Underneath there is a schematic neuron. And you see the dendritic EPSPs, they add up to form the spike. That's how we learned it in basic neuroscience. But then you also can have things that's called a dendritic spike, because dendrites can generate spikes. And you can have a back propagation. So if you have a spike that forms in the cell body, some of the electricity goes back into the dendrite. So it's believed all these things somehow come into the LFP, but exactly which contributes to what extent to the LFP is not really known. So the question is like, why I'm telling you this, like, what do we compare to our laminar data? Because in papers you see there's many different ways to analyze these neural data. So you have the raw LFP. In this case, they say the gammas, they associated with feed forward. So, and that tends to be in layer 4 here, the normalized power, or you can measure the neurons individually. That's like the depth summary. And another one is the visually the evoked LFP, and the derivative of that is the current source density. So if you see some of these power metrics, they go over, they're integrated, but the visually evoked and the CSD, if you see, they're only the first 100 milliseconds. So oftentimes people compare the fMRI signal with the CSD, but be aware it tends to be very short response. And the short onset response tend to also be much stronger than the sustained response. So we see with fMRI, because of the temporal resolution, we tend to see more of these sustained responses. But I just wanted to illustrate there's quite a variety of different neural metrics that you have. And this one I'm going to spend a little bit more attention on, because that's what you hear in the laminar field a lot, is that gamma is feed forward and beta is feedback. There's some evidence for that hypothesis, but as with the brain, it's all not always that straightforward. It's very nice and simple hypothesis, but it's just not always that straightforward. And that's what you see on the spectrograms on the right. We see that the power and the gamma in the upper layers, this is, and if you see, it's normalized power again. And you see the beta in the deeper layers, which, yeah, that's what they see. But look at the scale bar. The power in beta is much lower. The changes in beta is quite low, and that's the same you see on the right. You see the gamma tends to be layer 4 and up, so feed forward, and beta is lower. So that's this hypothesis. But on the other hand, there's also people like thinking that's maybe, is that just a property of the circuit, a property of these pyramidal cells and these shapes? Is it actually functional, or is it a byproduct of their anatomy? So, and this is what also in humans I wanted to show, that the laminar electrophysiology, and then in an attention test they also see gamma in higher upper layers, beta a bit in deeper layers. So that's also one of the remaining questions. What do these bands signify, and do they correlate with an fMRI signal or not? And this one I haven't talked about, I won't talk, but just mention it so that you're aware of it. There's neuromodulation involved in neurotransmission, neuromodulation is involved in behavior, a lot of psychiatric diseases, so it's pretty much everywhere. But one thing you see is that the receptors differ in the different layers. So we can fully expect that neuromodulation will have a laminar signature, but there's not been any much experiments in fMRI that I know of, but I would fully expect it. And this is also what Robert said about the different techniques. I've listed a whole lot of techniques in both neuroscience and fMRI that you can use to look at the cortex, and you see there's a huge number of them. And I don't think there's a ground truth in that. You have every different technique see something different. And this is from my vision course, where you see some animals see color, some see UV light. You look at the world in a different way, and everybody has different information to add, but there's not really one is better than the other. So as a summary, just to end the lecture, where does it leave us? So we have established the feasibility of fMRI. It's quite well established. And as you see, that's in the pictures on the right. So it does open up the possibility to study the circuitry. And the question, what exactly drives that signal, remains current. Also, we have plenty of technical challenges, and that's what all the other speakers will talk about. And but it might help if we understand the cortical anatomy, the function, how it functions. Then we can maybe ask the right question, help us design better stimuli, and it might improve our interpretation. And I think that's one of the things I do. I think there's always going to be a place for multimodal research, both in humans as in animals, because in humans it's starting to combine EEG with fMRI, and I think that's very exciting, and also in animals, because I think the answer of like what drives it is also only going to be if you combine multiple techniques. Okay, so I just want to thank the people who did some of the experiments, and thank you for your attention.